class.
Okay, this is the mythology that we teach our students about how cells work. And we all know that it's wrong, but we need to find ways to work on what's right, which is something more like this. And we're particularly interested in integrating metabolic status with all these other components of cellular function. And the reason why I refer to 3.5 billion years ago is that back in the beginning of pre-cellular life, or as we say in America, 6,000 years ago, all you had was metabolic status. DNA, this is a very late evolutionary event, and it's clear that in the origins of life, anything to do with nucleic acids only mattered in the context of creating a more stable, heritable physiology. So the hypothesis that we've been pursuing for some time is that if we understood cells beginning at this level, we would have our hands on the fundamental rule set. And then we would get insights into cellular function and normal development and in pathology. And this is all in the context of trying to develop a view of stem cell medicine to try and understand what stem cell medicine is going to look like 30 years from now, instead of the primitive state that we have it in now, and to start doing these kinds of experiments that will move it forward more quickly. Now, in terms of metabolic status, where I'm going to focus is on the black box or redox regulation. That redox regulation is something that we all understand is very relevant to understanding cell function, but we also understand that it is a mess to understand. That you have on the left side all of these components of developmental regulation, signaling molecules, and genetics, and toxicants, and stressors, all converging on changes in redox state, and you have a whole series of different outputs, changes in cellular self-renewal, changes in vulnerability to stressors, death, differentiation. How does that work? How do you get all of these different outcomes, all of these different inputs integrated? We've approached this by studying two fundamental problems in cellular function. One is the balance between self-renewal and differentiation. One of the oldest problems in biology for any cell that has the ability to differentiate. So the idea is that by studying this, we can get our hands on some fundamental principles. We also study the balance between survival and death. The cells that we use are cells that are involved in building the central nervous system. We now know quite a lot about the stem cells and the progenitor cells involved in building the nervous system, and we have a rich toolkit with which to work. The cell that I'm going to talk about in the first part of this talk is the oligodendrocyte type 2 astrocyte progenitor cell, which was the first precursor cell discovered in the nervous system by Martin Raff, Bob Miller, and myself back in 1983. It's one of the best studied precursor cells in all of biology, and it's a cell that we can work with at the clonal level. We can work with it in pure populations. We can work with it in vivo. We can do all sorts of interesting experiments with it in many, many different areas of function. I'm also going to talk a little bit about neurons also. Now, what's got us started thinking about these metabolic regulators as high-end regulators of cellular function was the following observation. We were interested in trying to understand how to prevent cell death in response both to mugging, physiological stressors like glutamate, turmeric rhesus factor, and also in response to neglect, in absence of the growth factors that you need to control cell survival. And we discovered that you could modulate these aspects of cell survival with antioxidants, and this was all done now 15 years ago. And what's been nice for us is that what we consider to be the most important part of these studies was completely ignored by everybody else, which is not that you can use antioxidants to control cell survival, but that cells are completely dependent upon their redox state in order to even respond to extracellular signals. So here we're looking at the survival of neurons from the dorsal root ganglia, from spinal ganglia, and we put them in a tissue culture dish in minimum conditions, and then we try and keep them alive. In the purple bar, we're adding nerve growth factor. which is the best studied survival factor for these nerve cells. In the green bars, what we're doing is adding a chemical called N-acetylcysteine. N-acetylcysteine is an antioxidant formulation that has been in clinical use for more than 50 years. It is the treatment of choice for Tylenol, acetaminophen toxicity. It is used in treating cystic fibrosis. It's used in many settings. And back when we did these studies, we discovered that it prevented death by tumor necrosis factor and glutamate and other things. What we also found is that by giving cells N-acetylcysteine, we changed the response to other signaling molecules. And what was intriguing about that is that N-acetylcysteine is taken up by cells, it's converted to cysteine. Cysteine is the rate-limiting precursor for the biosynthesis of glutathione, which is the major regulator of antioxidant function in a cell. And if we increased glutathione levels by just 15%, we now got this 1,300% increase in the number of surviving neurons. So you put in $1, you get $80 back, much like all the economists are able to do for all of us. And as we went on to study this, we found that this applied to all the receptor tyrosine kinase agonists we looked at. It applied to all the cell types that we looked at, and it also applied to precursor cell function. And we found that by adding N-acetylcysteine to cultures of the oligodendrocyte progenitor cells dividing in their basal mitogen condition, we changed their differentiation. Now, these graphs look complex. They're actually very simple. What we're doing is we're taking cells and studying them as individual clones of cells and studying the differentiation of a clone of cells. And we plot this to show the number of progenitor cells in a clone in the back wall, the number of oligodendrocytes in a clone on this wall, and the height of a bar is the number of clones with any given composition. And all you need to know is that's lots of division and that's lots of differentiation. If we take cells from the optic nerve of a 7-day-old rat and grow them in the presence of platelet-derived growth factor, the basal mitogen, you get clones that differentiate and clones that don't. If we first purify these cells on the basis of their redox state and separate them into cells that are a little more reduced and a little more oxidized, what we then get is this, where the cells that are a little more reduced in the animal divide extensively. The ones that are a little more differentiated in the animal differentiate. They're not committed to differentiating because if we give them N-acetylcysteine, now they divide extensively. So the balance between self-renewal and differentiation is controlled by the redox state. At the same time when we're doing this, we're trying to figure out why different regions of the nervous system develop at different time scales. Why does the spinal cord myelinate early? Why does the cortex myelinate late? Well, we looked at the level of individual progenitor cells and found that the cells were very different. Grown in basal division conditions, optic nerve progenitors over the course of 6 days generate clones where almost every clone has oligodendrocytes in it. This is a tissue that myelinates early. The same cells shown at the clonal level to have the same differentiation potential, isolated from the same animals, but from the cortex, grown in the same conditions, don't differentiate hardly at all. And these differences are cell intrinsic, and as we went on to study this, we found that what appears to be the controlling factor is the intrinsic redox state of the cell when it's isolated from the animal. These cells are more reduced, and these stay more reduced even over the course of 6 weeks of growth, even in the course of growth in atmospheric oxygen. So the organism appears to be using redox state to control the timing of differentiation. So redox state controls precursor cell function. I'm going to get to classical signaling molecules a little bit later. Okay, so here's where we were. We had found that if you're more reduced, you have better survival. If you're more oxidized, you have more death. If you're more reduced, you have more self-renewal. If you're more oxidized, you have more differentiation. And we were able to figure out some rule sets that apply for this lineage. If you're more reduced, you're more responsive to mitogens and survival factors. If you're more oxidized, you're more responsive to differentiation and death factors. So for someone like me who's interested in tissue repair, this is a disaster because tissue injury is an oxidative stress situation. and you're enhancing the response to exactly what you don't want to enhance the response to.